Hello and welcome to today's photosynthesis webinar. I'm Louisa Diva and I'm presenting from a beautiful office in Amsterdam today. I'm very pleased to present to you our two guest speakers, Professors Zoran Nikoloski and Paolo Pesaresi. I should remind you that the webinars are sponsored through the Capitalize programme, which is part of a cluster of European projects that are exploiting photosynthesis for crop improvement. Okay, so we'll move on to our first speaker today, which is Zoran Nikoloski. He is the chair of the bioinformatics at the Institute of Biochemistry and Biology at the University of Potsdam. He's also a collaborative research group leader at the Max Planck Institute of Molecular Plant Physiology, and his research is involved with understanding how genomic variation leads to different metabolic phenotypes and growth-related traits in photosynthetic organisms. He's a member of the Capitalize project, where his team is focusing on modelling natural variation associated with Calvin-Dancer cycle, as well as integrating data from high-throughput phenotyping to predicting genotypes with improved photosynthesis. Today he's going to talk to us about machine learning of photosynthesis-related traits using hyperspectral reflectance data. Hand over to you.
Thank you, Louisa, for a kind introduction. So I'm going to introduce part of the work which we have done in the context of the Capitalize project. And in the context of Capitalize, we are interested in understanding major drivers of photosynthesis. We're interested in photosynthesis because this is the basic process by which solar energy is converted in plants into chemical energy. And this chemical energy is then used to fix carbon dioxide into sugars and other building blocks of biomass. And as a result, photosynthesis has a very important impact on the carbon cycle on our planet, but also it's very important for understanding what drives the productivity of crops, especially if we are interested in products which are directly related to biomass. As part of photosynthesis, we also have production of oxygen. So this process is the key oxygen source in our atmosphere. And as a result, due to all of these properties, we can say that photosynthesis affects most of life on Earth. Now, photosynthesis has been studied for decades, and most of the work in photosynthesis has been done experimentally. However, there is another way of studying photosynthesis, and that is based on modeling. So when we want to start and model photosynthesis, we certainly have to first understand what are the underlying processes that are key to this process. So photosynthesis can be divided into 4 steps. The first is light absorption, in which different types of pigments sense and capture light in coordination with different proteins that lie in the thylakoid membrane. And as a result of this, they basically transfer the energy and contribute to splitting of electrons from electron donors like water that contribute to the production of oxygen. These electrons are then transferred in a series of reactions that contribute to the production of NADPH, which is one of the key energy currency in a plant and in any other organism. As part of this process, we also have the creation of the proton gradient, and the proton gradient is then used by the ATPase to produce the other energy currency, which is ATP. Both of these energy-rich molecules, NADPH and ATP, are then used in the Kelvin-Benson cycle, which is the key biochemical cycle, in which the enzyme Rubisco, the most abundant enzyme on planet Earth, fixes carbon dioxide in interaction with ribulose bisphosphate, RuBP. This Kelvin-Benson cycle process can be divided into 3 stages of carbon fixation, reduction, and then regeneration of RuBP. And as key aspects or key products which are generated from photosynthesis, we have the sugars and other types of molecules that then contribute to the creation of building molecules of biomass. So of course, this is the essence of photosynthesis as described by a modeler. But of course, we have different types of photosynthesis across plants. We study two different types of photosynthesis in the context of capitalize, and we try to understand what is the natural variation that exists for different types of photosynthesis related traits in organisms that in plants which have C3 photosynthesis, such as barley and tomato, in which the Kelvin-Benson cycle runs in the mesophyll cell, and then maize as a representative C4 species in which photosynthesis is divided between two cell types, mesophyll cell and bundle sheath cell, in which the Kelvin-Benson cycle takes place. Clearly, the modeling of photosynthesis is going to differ depending on the two different types. But if we start with the simpler model of the C3 photosynthesis, then can later be extended to C4 photosynthesis. So this is why for the rest of the talk, I'm only going to focus on simple basic models that have been used to study C3 photosynthesis. One of the basic models is a steady state model, which was proposed by Fakwa, von Kammerer, and Berry already in the early 80s. This is a very important model because it allows for prediction of net carbon dioxide assimilation, which is denoted by A, not only on a level of a single leaf and single plant, but also can scale on the level of canopies. According to the model, the net carbon assimilation rate can be described by the minimum of the Rubisco limited rate of carbon dioxide assimilation, which is denoted by AC, and the electron transport limited rate of carbon dioxide assimilation, which is denoted by AJ. If we now go through the mathematical descriptions that have been proposed for the two different rates of carbon dioxide assimilation, we will realize that these depend on several parameters which are kind of fixed for a genotype, and things that may vary across the genotypes and are determined by the turnover number of the respective enzymes, as well as the abundance of the active enzyme. One of these properties is the maximum rate of carboxylation of Rubisco, which is denoted by Vc max, and the other one is the J-max, which is the maximum rate of electron transport for ribulose bisphosphate regeneration. Clearly then, if we have this equation and we have values for the parameters, we have the capacity to predict based on these equations what the net carbon dioxide assimilation rate is going to be in a particular plant, in a particular genotype. The problem then is how to obtain the values for these parameters. And the values of these parameters are obtained by fitting so-called photosynthetic carbon dioxide response curves, which are also denoted as ACI curves, that are obtained from gas exchange measurements. So you can imagine that these measurements are quite costly and quite time consuming. So as a result, if we would like to be able to understand natural variation in net carbon dioxide assimilation in a population of genotypes, then the bottleneck clearly is obtaining measurements for these two key parameters that would then allow us, using the equations that I presented, the prediction of net carbon dioxide assimilation rate. The problem then is how to arrive at facile means to obtain the values for the two key parameters that would allow us to generate genotype-specific models. One obvious solution would be to use data from high-throughput, non-invasive phenotyping, and to use this data with machine learning approaches in order to predict values for Vc max and J max, because then once we have the machine learning predictions about the two values, we can plug them into the mechanistic model of Fakwa, von Kammerer, and Berry. Now, what are some possible high-throughput non-invasive phenotyping measurements that we can perform? One of them is given by leaf hyperspectral reflectance measurements, we are in a good position these days because we have such handheld devices which, provided a sample leaf, have the ability to generate such a reflectance spectrum for a given leaf, which capture how much percentage reflectance is being emitted at different wavelengths, which are shown on a leaf. So the idea is then to use precisely the values which we have of the percentage reflectance as predictors over the different wavelengths with the different response variables which we are interested to model, one of which are VC max and G max. The main questions that I will try to address in today's presentation are threefold. The first is, can we actually predict different traits, including photosynthesis related traits, by using hyperspectral reflectance data? The second question is mostly related to methodology, because we want to understand if this can be done by using machine learning approaches, which machine learning approaches outperform others, and do we have any differences between species? And then the final question is, can we show that the models can generalize to genotypes that we have not seen in the actual training of the model, as well as generalization of the models across growth seasons, because it will tell us that indeed we can rely on these predictions if the models are good enough. So we are going to go step by step in addressing these questions, and to address this question we use data which are obtained in the context of capitalized project. We rely on two populations. I'm going to present only results for maize and barley. For maize, we rely on the maize magic population, which is obtained from Matteo Dell'Acqua's group at Sant'Anna. This population was grown in 2021 and 2022 with 626 and 633 genotypes from the magic population, allowing us to address the third question, the between-year predictions and generalizability of the model. And the second panel which we use is the barley diversity panel from A.L. Friedman's group at the Volcani Institute in Israel, which was grown in 2022 with 509 genotypes. For all of these, we obtained the leaf hyperspectral reflectance. This was work done in the group of Johannes Kromvik at Cambridge University. And in this group, there were also a number of traits that were measured. I'm going to divide the traits into so-called physiological traits that capture specific leaf area, percentage carbon, nitrogen, and isotope-related traits. We then have traits that are obtained by fitting the parameters using the ACI response curves, and these are precisely the VC max and J max that entered the model of C3 photosynthesis. We have light saturated gas exchange rates, and finally fitted parameters using chlorophyll fluorescence, which are related to non-photochemical quenching and efficiency of photosystem 2. For every genotype, there were measurements obtained from 2 plots on the same field, each with at most 3 replicates, which also allowed us to ask questions about prediction of values for one genotype across plots, as well as for genotypes that are not seen previously within the same season. So this data clearly are very rich and allow us to address the 3 questions which we started off with. But I mentioned that we have a methodological question that deals with the machine learning aspects of the work. So what is the methodology that we base this research on? So clearly there are challenges which are related to learning models from hyperspectral data. Clearly, we have many more predictors, in this case, reflectance at different wavelengths. Then we have samples, which are the measurements across the different genotypes. Many of these predictors show multicollinearity that is also related to overfitting of the models which we obtain. From data science, we know that one possible solution to the mentioned problems is to rely on machine learning methods that include techniques based on regularization as well as feature selection. As a result, after running multiple analyses, I'm just going to present the comparative analysis of three approaches. The first one is partial least square regression, which is commonly used in all of the previous studies that rely on prediction of various types of traits from hyperspectral reflectance data. We have support vector machine and elastic net regression. Elastic net regression, like partial least square regression, is a linear model, where support vector machine, by the way of using radial kernel, allows for capturing nonlinearities as well, and allows us to also address questions about whether nonlinearity helps with the predictability or increasing the accuracy of prediction for the traits that we modeled. Tightly related to the issue of overfitting is the issue of cross-validation. So we perform two types of cross-validation. The first one is done for the purpose of performance assessment, and this is done in three scenarios. In scenario 1 and 2, we are dealing with model learning within with data from a single season, from a single year. And there we distinguish the scenario of predicting seen genotypes by using data from different plots. And then we consider the more difficult scenario of prediction of unseen genotypes. And of course, the most challenging scenario is the one where we train the model on one season and predict the values for the traits measured in another season. To be able to assess the performance of a model, there are different ways in which we can go. We decided for this particular purpose to present the values or to present the performance in terms of coefficient of determination, which basically tell us how much variance is explained by the model. In addition, we perform hyperparameter tuning in order to make sure that we present those results which are most optimal. And here for visualization, I wanted to illustrate that indeed we make sure that we try to minimize the data leakage issue that occurs and often results in overfitting of the resulting models. So with data explained and methodology explained, I move on to presenting the results. I will start off the presentation of results by focusing on barley data. And I want to remind you that in the case of barley, we only had measurements from a single season. So as a result, for barley, we can only treat the scenario 1 and scenario 2. In scenario 1, we are predicting seen genotypes because we are using values of one genotype in one plot to predict the values of that genotype in another plot on the same field. And you realize that I have divided the results into 4 sections. The top left presents the results from modeling of the physiological traits. On the top right, you will find the results from the parameters obtained by fitting the ACI curves. On the bottom left part, you will find the parameters obtained by chlorophyll fluorescence. And finally, on the bottom right, you see the light saturated gas exchange rates. With different colors, I show the performance of the 3 methods which are being tested, PLSR, SVM, and GLMNet. And if we now go a little bit deeper into these results, we will quickly recognize that the best predicted parameter, which we have, the best predicted trait, is specific leaf area, and that this is matched by the carbon to nitrogen ratio, followed by the nitrogen percentage and carbon percentage. All of the others are more or less hovering at 0. Interestingly, with respect to having the ability to predict photosynthesis for unseen genotypes, we also realized that the current models which we have across the linear and nonlinear that we are comparing, we don't see much of a difference, and we can explain about a little bit over 30% of variance on average for both VC-max and J-max that enter the mechanistic model. The same holds for some of the other parameters which are obtained by fitting of ACI curve, but we realize that yeah, for some of the predictions we also have overfitted models or poorly fitted models that result in R-square values that are below 0. Similar situation is found for the chlorophyll fluorescence parameters, which are poorly predicted by hyperspectral reflectance data. And from the light saturated gas exchange rates, the best prediction is obtained for saturated net assimilation rates, and we do not have good predictions for stomatal conductance, for intercellular carbon, as well as for water use efficiency values, which are obtained from these gas exchange rates. If we move on to the second scenario, which is the one of unseen genotypes, we expect that the results cannot get better, they can only get worse because the prediction scenario is more difficult. And indeed, this is what we observe. Interestingly, for the physiological parameters such as SLA, specific leaf area, as well as for carbon to nitrogen ratio and nitrogen percentage, the results do not really differ in comparison to the scenario what we've seen for the seen genotypes. However, for VC and Gmax, we already observe a third decrease in accuracy in comparison to the scenario with seen genotypes that are presented on the previous slide. The rest of the parameters I do not go in detail because they only become more poorly predicted. So having seen such performance, which is not very striking for barley, we were wondering whether at all the situation is better or worse in the situation of maize. If we turn to maize, we realize that the prediction for maize is drastically better, particularly for specific leaf area, for carbon to nitrogen ratio and to nitrogen percentage. So similarly to what we saw in barley, now we even have larger accuracies which are obtained. In this case, VC max is actually even better predicted in comparison to barley. For the other parameters, we have similar performance as in the case with the barley diversity panel. Unseen genotypes, situation is similar. We observe a decrease, but not as big a decrease in performance, which actually tells us that if we have an extension of the mechanistic model of C3 photosynthesis to C4 photosynthesis, we are going to be able to actually do a nice genotype-specific prediction of net photosynthesis rate. The most difficult prediction scenario is that of prediction across years. And on these graphs, I try to capture simultaneously the training scenario where the model is being trained on data from 2022, and then it's used in prediction of a model diagonally opposite in a testing year, which is 2021. And you realize that in the case of the specific leaf area, the correlation drops from 0.89 to only 0.84, which means that the models that are learned in the case of maize are highly generalizable across year for this particular trait, which is again confirmed if we do the reciprocal prediction, if we train a model on data from 2021, where we have a correlation of 0.9, that correlation testing data from 2022 to 0.74, which again gives us a confirmation that the models which are obtained from maize are actually more generalizable. While this is a parameter on which the models perform quite well, we have other scenarios and other traits, like for instance the saturated net photosynthesis rate, where we have actually dropped by about a third in the correlation from the training to the testing year, as you will observe from the correlation which are shown diagonally. So of course, we are not the first to do this type of analysis. Leaf hyperspectral reflectance data have already been used for prediction of photosynthesis related and other plant traits. So we also attempted a comparative analysis against a seminal study from Jendrek et al. From plant physiology, because from this study we could both obtain the approach that they use, which is the partial least squares, and also we had access to the full hyperspectral data, and we wanted to do a comparison whether our data are better in capturing variance in comparison to what is published in that publication, simply probably because of the type of genotypes which we are using, or type of material which we are using. And you can appreciate from this table, you realize that the prediction which we obtain for some of the key parameters for photosynthesis, as well as for the best predicted physiological trait, we actually increase the variance explained by the strategy that I presented here in comparison to what was used in the respective paper, which shows that the methodology which we have chosen is on par with what is classically used, but also the setup of the experiment and the usage of the maize magic population, it's quite valuable in capturing the variability nicely by the machine learning models that in the next step will be integrated into mechanistic models of photosynthesis. So in conclusion, we learned that the models which are generated based on hyperspectral reflectance data can better predict photosynthetic related parameters in maize than in barley. We also showed that the developed machine learning models generalize between years and also outperform contenders. And finally, because of the introductory presentation which I did of the tight link between the machine learning prediction of parameters and the mechanistic model of photosynthesis, these type of models now pave the way for genotype specific mechanistic models of photosynthesis and a way of basically screening larger population of genotypes without the need of doing the laborious gas exchange measurements. So with this, I'd like to thank the people who were largely carrying this work. I would like to foremost thank my PhD student, Rudang Su, who did the work in collaboration with a postdoc, Mona Razagi, hired on the Capitalize project. I would very much like to thank the collaboration with John and Wane from University of Cambridge, who provided all of the data. I'd like to thank the entire group for the support and the funders for supporting this line of work. With that, I would like to thank you for your attention, and I'm open for any questions.
Thank you very much, Zoran. It was lovely to see a presentation on modelling that wasn't full of equations. Quite a relief. We have a question already. So please put your questions in the question box. We have one already. Why is prediction better in maize than in barley? Is it due to the type of population used?
We suspect that it is the population used, correct? That we have larger variability in the scenario of maize in comparison to barley. And this variability together with, we need heterogeneity to be present. in order to be used for learning of the models. But of course, we don't like also too heterogeneous data. So in the case of barley, we also had some outliers that also contribute to the problems with learning of the models. So yes, my answer would be it is the type of population which we are using.
Okay. And how difficult is it to include stress parameters into your modeling data? Because presumably, you know, different genotypes respond differently to stresses. You know, if they've got thicke cuticles or sunken stomata for whatever reason. So it might look like it's performing really well under an optimal year, but not so well, or the other way around, not so well in an optimal year and better in a drought, for example.
Yeah, so this type of modeling that I presented here goes under the umbrella of phenomic prediction, because we're just predicting phenotypes. In this case, the phenotypes are the parameters of the enzymes that enter the mechanistic models. Clearly, phenomic prediction models do not only rely on hyperspectral reflectance data, we can also include environmental variables as part of the prediction. So as long as metadata about the environment at the moment of gathering the phenotypes is also collected, then this can definitely be integrated readily as just another predictor, which can even be weighted more strongly in comparison to others in explaining the variance of the phenotype which we are modeling. And actually, there are quite a few modeling attempts, particularly in the field of quantitative genetics, that go towards prediction of end plant phenotypes like yield by not only considering genotypic values, which I do not focus on today. It's phenomic prediction based on hyperspectral data. By inclusion of environmental variables. So yes, this can be readily done as long as we have such data at hand.
Yeah. Although I suppose it's infinite how many environmental variables you could include, you know, like soil, nutrition levels, or water levels, or all sorts, can you really?
Surprisingly, there are databases that have been collected in the last 2 to 3 years, particularly for maize, for rice, and for soybean, where actually we have variety of information about the enviromics, as they call them, where we precisely capture information about soil properties, watering regimes, light quality, temperature, and so on and so forth. And they have been already used in various modeling attempts.
Okay, thank you. There is another question. What are the most predictive features of the hyperspectral data? Can they be related to biochemical compounds or morphological characteristics?
That's a beautiful question. Yeah, I did. In interest of time, I could not go in the direction of presenting the importance of features. And indeed, if we do that exercise, which we have done, one would realize that the portion of the spectrum that is responsible for pigments, that is responsible for protein, that is responsible for nitrogen, sugars, and so on, these are the ones that are basically picked up by for the particular phenotypes that we're modeling. Of course, they differ from model to model depending on the phenotype which we're modeling. But for instance, in nitrogen, you realize that it's the picked which is related to nitrate. For carbon, you realize that is partitioned part towards the carbohydrates, which are on the bottom right of the spectrum, and related to the protein content in the middle. So yeah, these are typical patterns that one would expect actually arise, which was quite nice to see, because it's a confirmation of the informative value that this type of data have. But spot on question, thank you for it.
So in the interest of time, I think we're going to move on to our second speaker, but if there's any further questions, we may have time at the end to answer them if they're in the question box. Okay, thank you, Zoran.
Thank you.
We're moving on to our second speaker, which is Professor Paolo Pesaresi. He's a full professor of genetics at the University of Milan and leads the Photo Lab group. He's interested in short and long-term regulation of the light phase of photosynthesis and in chloroplast biogenesis using functional genomic approaches in Arabidopsis and in barley. His previous work has included identification and characterization of genes involved in regulation of photosynthesis. Now, he's currently leading the recently funded EU project called Best Crop. And today he's going to talk to us about how Best Crop is boosting photosynthesis to deliver these novel crops for the circular bioeconomy. Over to you, Paolo.
Thank you, Luisa. I would like to thank, first of all, Capitalize to give me the opportunity to introduce the main objectives of the Best Crop project that has been recently funded by European Commission and in part by the UK Research Innovation Agency. Just to give you some numbers, the Best Crop project is made of 18 partners, 10 are university and research institutions, whereas 8 are companies, specifically company of the breeding and seed sector, and also company involved in producing bio-based compounds and material. It will be a project of 5 years and has been supported with 6 million euros, and the project started the 1st of July 2023. We are just at 6 months of the project, so I will not have many data to show to you. The purpose of this webinar is just to tell you a little bit about the experimental plan and the main objectives of Best Crop. Actually, Best Crop has the ambition to develop a next generation barley plants with the 3 main characteristics. These barley plants should have an increased ability to uptake carbon dioxide and ozone, should have an increased production of above ground total biomass by 15 to 20% without touching the harvest index. And in addition to these 2 features, the new barley plant should have a tailored straw that could be used by companies working in the bio-based circular economy. And in particular, the final aim is to use the low-value straw barley to produce high-value bio-based compounds like biolubricants, construction or insulating panels, as well as a feed for animals. The Best Crop work plan is organized in 9 work packages, and the first part, the first 4 work packages are dedicated to the identification of barley lines with specific traits, to pyramiding together these traits, and to test the barley lines under first control condition, greenhouse condition, and later on bring a subset of them into the field and test their performance. In particular, concerning the first work package, we are interested in isolating lines with improved photosynthetic performance. Our specific aim here is to improve the light ability of barley canopy to absorb light, and we aim to do that by reducing the antenna size of barley leaves in order to allow a better penetration of light towards the canopy, therefore reducing the amount of leaves that are constantly under shadow and that do not almost perform photosynthesis. We also aim to obtain lines that better adapt to the very rapid changes in light intensity due, for instance, as you can see in this movie, by the movement of the plants caused by the wind. And we are trying to do that by increasing the kinetics of non-photochemical quenching and by also introducing flavodiiron proteins. We also aim to modulate stomata opening for increasing the uptake of CO2 and ozone. And in addition to that, we would like to also to have some transgenic lines where we introduce a synthetic photorespiration bypasses. This is just an example of the lines we will work with, and many more will be identified during the first phase of Best Crop project. This is just an example of those lines with the reduced antenna size that will allow a better penetration of the lights towards the canopy. And this is, for instance, an example of line where we change the canopy architecture. In particular, we introduce directly phenotype in order also to reach the same objective, that is a better penetration of light towards the canopy and more uniform photosynthetic performance towards the entire plant. Many more pale green mutants will be identified by exploiting the many lines available in this Chlorina mutant collection that is made available by Matt Hanson at the Lund University. We are currently screening them for their photosynthetic performance and for their leaf chlorophyll content, and try to isolate those ones that show an improved photosynthetic performance and a different degree of chlorophyll content reduction. In addition to that, we are running also some tilling analysis on candidate genes involved in determining the content of chlorophyll in leaves, genes involved in non-photochemical quenching, in ozone uptake, in leaf erectness, and also in the cell wall properties. And we are exploiting mutant population in barley in the background golden promise and laureate that are provided by our partner at the James Hutton Institute, and specifically the group led by Robbie Vogt. Besides this induced genetic variability, we are also exploiting natural genetic resources in barley. And again, our partner at the James Hutton Institute made available the Kranakan database that contains exon capture data on more than 1,000 barley lines, and these are related to wild barley, to land races, but also to elite germplasm. And also this allele mining will be run for the same traits, chlorophyll content, non-photochemical quenching, etc. The third approach will involve genetic engineering, and in particular gene editing, and also the introduction of synthetic pathway. And we will do that on elite barley varieties, specifically on Carbona. And here we will try to generate lines carrying new genes or overexpressing new genes, but also try to get knockouts in specific candidate genes. One of this genetic engineering strategy will be used, for instance, to introduce, in collaboration with our partner, University of Padua, Thomas Morosinotto, to introduce a flavodihyron protein taken, for instance, from Fiscomatrella and bring it into higher plants. And there are already data in model organisms that clearly show that the introduction of those flavodihyron proteins make plants better adapted to fluctuating lighting condition and even under drought condition. As I said, the other genetic engineering approach that will be used to introduce, in collaboration with our partner at University of Düsseldorf, Andreas Weber, to introduce novel photorespiratory bypasses based on also in part based on existing knowledge that have been recently published. And finally, also, we will try, as I said, to modulate the opening of stomata by acting on specific protein pumps, and by selecting specific mutants, especially mutants in this domain that is the regulatory domain of this protein pump. Our aim is to identify lines that will maintain stomata open, but at the same time it will be responsive to drug and to abscisic acid stimuli. And this has been already shown, this approach to work in Arabidopsis, where line mutated in this specific protein pump are able to produce more biomass, but at the same time, and therefore absorb more CO2 and ozone, and at the same time also being able to be responsive to drought and to abscisic acid stimuli. This is the first part, the one dedicated to improve the photosynthetic traits. And as I said, then this increase in biomass is coupled with an improved quality of the barley straw. And in particular, we will try to exploit the relatively large natural genetic variability that is present in the barley germplasm concerning lignin content, for instance, and lignin properties. But also we will exploit many different transgenic lines developed in the lab of Claire Alpin at the University of Dundee, where she downregulated the expression of several genes, for instance, involved in the lignin biosynthesis. And she was able to obtain many lines with different content of lignin and also different lignin properties. And another aspect that we will try to address during best crop would be the amount of protein in the barley straw. Also here, we will try to exploit the natural genetic variability concerning the percentage, the amount of protein into the straw. This is a collection, for instance, that have been provided to us by ICARDA, and they were able to select by screening around 200 different genotypes to select lines with a certain, with a high amount of protein that is closer to around 6%, and also lined with very low amount of protein. So this in particular, obviously the line with a higher content of protein straw will be combined with lines with an improved photosynthetic performance, and also with lines that has a specific lignin characteristics. All these traits in work package 3 will be then stuck together according to this breeding scheme, and this will be done in collaboration with our research partner at CREA in Fiorenzuola d'Arda here in Italy. But we have, as I said at the beginning, several companies working in the breeding and seed sector, and they will be instrumental to implement all our breeding strategy. The line, as I said, will be also checked under controlled condition in greenhouses, and a subset of them will be then moved to the field. This is, for instance, one of our first field trials where you can recognize one of our pale green mutants that are grown together with the corresponding control. The second part of our project is dedicated to transform then the barley straw of these selected lines into bioproducts. This is actually the aim of work package 5 and work package 6. And in particular, after selecting the lines with the specific cell wall composition and physicochemical properties, we aim to obtain for barley straw proteins as additive for animal feed, but also oleochemical products. But at the same time, straw will also be used to build insulation and construction panels. This will be done by different industrial partners that we have in our project. For instance, Sogis, that is a company's leader in the sector of oleochemicals, will use barley straw in combination with black soldier flies to produce proteins and biolubricants. Mogu will use the barley straw in combination with mycelia to obtain insulating panel, whereas Mogu, and in collaboration with FR Decodem and the Colmine Telecom will be also use barley straw using different strategy to obtain construction panel and polymer composites. Just to give you some details about the strategy, for instance, we can use barley straw after identifying the specific lines with an increased protein content to use these lines as feeding for our black soldier fly larvae, and these larvae will be then used to obtain from them, after pressing and enzymatic treatment, oil and hydrolyzed protein. And later on, oil will be again split into fatty acids and glycerol, and proteins can be used as an additive for animal feed, whereas fatty acids will be used for the production of biolubricants. Mogu will produce, as I said, insulation panels, and the strategy they use is simply to take barley straw, to let the barley straw colonize by mycelia from selected Ascomyceta and Basidiomyceta, and after colonization, they will mold the barley straw into the shape they wish. Then the mycelia will be simply inactivated by autoclaving them, and then the mycelia will work as a natural bending agent, and this will lead to the construction of acoustic and insulation panel. With different strategy, our partner FRD Codem will also be able to obtain from the barley straw panels with improved lightness, mechanical performance, and also with improved ability to resist to fire. This will be the main bioproduct that will be obtained in our project. Of course, we will run demonstrator, and this will be at year 4 and year 5. We will have demonstrator concerning our lines obtained by exploiting natural genetic diversity, but also exploiting mutant plants obtained by chemical mutagenesis. And they will be grown in fields that we plan to have in the north of Italy, in Fiorenzuola d'Arda, exploiting the facility of our partner at CREA. We will grow them in the north of Germany at the KWS facility, and we will also grow them in Denmark thanks to our partner Nordic Seed. As I said, we are also exploiting gene editing and genetic engineering technology, and we will grow these lines in the facility that is made available in the Czech Republic by Uzovsko, where they have a specific license to grow genetic modified plants under field condition. And the same will be true for our company that will be dedicated to transform the barley straw into the product that I mentioned, into biolubricants, into insulating panel, into construction panels. And there will be in the north of Italy, Sogis and Mogu, but also FRD Codem that is placed in France. And finally, we will also try to use the scenario that is present in the north of Italy, where we are going to grow the barley straw in Fiorenzuola d'Arda and transform the barley straw at Sogis and Mogu, here producing proteins and biolubricants, here producing insulation panels. And this would be our ideal scenario to predict, to assess the life cycle of this strategy, and also the technical and economical feasibility. Idea, let's say this approach in order to be sustainable need to have a scenario like this, where you produce the straw in a certain place and not far away from it, actually very close to it. You are going to transform the straw into those products. This is actually our main aim, and these are us at our kickoff meeting here in Milan that we had in July 2023. You can find us at the moment on Facebook, LinkedIn, and Twitter, and very soon, at the beginning of January, we will also have our website where you can find further details of the BEST project. And with this, I finished, and I'm pleased now to take some questions.
Thank you very much. That was a really interesting talk about your project and all the translation of the work you can do with it. Everybody else, please put your questions in the question box. I can put to Paolo. I've got a couple already, actually. I'll have some myself. The first one asks, what is the relationship between FLV and NPQ?
Okay, so the FLV is a final electron acceptor. So let's say it can be, it's not present in higher plants, but there are studies, even in barley, that show that under environmental, very rapid changes of light condition, but even under drought stress, it can work as a sink of electrons and therefore reduce oxidative damages. And the non-photochemical quenching works in a different way. The non-photochemical quenching is actually removing the excess of energy as heat, but the final goal also is to prevent oxidative damages. So essentially are two different mechanisms, but with the same aim, that is to protect plants from the excess of energy or the excess of electrons at high energy.
Okay, thank you. Why did you decide to work on barley? Why not maize?
Well, I believe barley, it's becoming an ideal model crop for photosynthesis. It was in the past. When I started, I remember my master thesis, I was working with some barley mutants. Many of the barley mutants that allow to discover, for instance, a chlorophyll biosynthetic pathway, many other major discoveries in photosynthesis have been done in barley in the 80s of the last century. Then came Arabidopsis, of course, very simple model, very with a very rapid life cycle. The genome is easy to transform. But nowadays we have an excellent barley genome. We have a version 3 of the Morex assembly that is, I would say, almost perfect at the level of the Arabidopsis genome. Barley can be transformed. We have a huge collection of mutants, but what is most important is that you can grow barley from the pole to the equator. So you have a tremendous natural genetic variability. These are the main reasons why we decide here to focus on barley, obviously with the final aim then to translate the knowledge into other, let's say, more important crops, for instance, wheat.
Okay. Another one, wouldn't you also consider doing some work on assimilation since you're aiming to increase CO2 uptake?
It's not really the main aim of this project, but certainly it's a very important aspect. We do not have this activity in best crop, but I fully agree that looking at the product of this increased CO2 assimilation would be certainly very interesting.
So I've got a question. So you talk about your 4 improvement strategies for photosynthesis: the antenna size, the NPQ, kinetic, stomatal, and the photosynthesis bypass. Are these chosen for a particular reason? So you're looking at wanting increased biomass as well as increased grain yield. Is there any indication of which of these improvement strategies will give you biomass versus grain yield, or is that not known yet?
Let's say most of the proof of concept that show that, for instance, a pale green phenotype can increase the total biomass or even the NPQ, or even increasing the kinetics of NPQ can lead to an increase in photosynthetic performance. Most of this research has been done on model organisms and has focused mainly on total biomass. There was not a specific, for instance, concerning the NPQ, we have very interesting studies on tobacco. For the pale green phenotype, there are some very interesting studies on soybean, but none of this study focus on the grains. So let's say we don't know now. I would say in one study on the pale green phenotype that I conducted before starting this project, done on a pale green mutant, probably is one of the first studies where able to show that when we touch NPQ, when we touch the pale green, the leaf chlorophyll content, we don't have any major defect in the grain yield, in the harvest index. But this is an aspect, actually, that's one of the reasons why we select barley also. It's a time to look at the impact of this strategy, not only on total biomass, but also on the grain yield, and therefore at the level of the harvest index. I can make some prediction. We don't expect much changes, but I think it's time to prove it under real field condition, and that's why we would like to use barley. Yes.
Are you using heritage barley then as well? Because presumably you're looking for tall stems, or is that not an issue? Are you not looking at tall, long stems for this work?
No, we are not really using barley with a tall stem. Actually, as you have seen in our breeding program, the idea is to use the lines with the tail or a barley straw as a recurrent parent, because obviously it's relatively easy to identify the traits that allows you to have a reduced chlorophyll. Much more complex are the traits that control cell wall. So we will use this as a lead material and as a recurrent parent in our breeding scheme. But there we don't have a very tall stem, very tall culm in there. No.
Okay. Well, thank you very much. I'm afraid due to time constraints, we'll have to bring this to a close. So it just leaves me to thank our speakers for two really stimulating talks. Thank you very much. I really enjoyed them. I hope our audience did as well. Thank our audience for attending. And finally, remind you that we have our next webinar in February, on February the 6th. This will be featuring Dr. Lamis Abdel Hakim, who works at PSI and is a member of the Capitalized Consortium, and using some of the instruments at PSI to phenotype barley again, looking at resilience to multiple stresses. So it's open for registration now, and I look forward to seeing you then. Thank you to the speakers, and thank you to you all. Goodbye.
Thank you, Louisa.